Hi, my name is Elizabeth Villa, and I'm along with Bridget Caraher and Ilaria Testa. I'm one of the co-organizers of the Keystone Symposia themed Imaging Biomolecules Across Scales from Atoms to Tissues that will take place in March in Keystone, Colorado. The idea behind this meeting is that there's a lot of techniques that are coming together and sort of meeting at trying to look at the complexity of the cell at ever-increasing resolution, both temporal and spatial. And so we're trying to have people from different techniques get together and just talk about what their challenges, what their opportunities are, and how we can really come together to try to get to see biomolecules in action at exquisite detail inside cells. The program is centered in methodologies, and it really goes from conventional, quote-unquote, structural biology, then AI, all across the scales to cryo-electron tomography, volume, correlative light and electron microscopy, and all the way to like super fast and better light microscopy that really allows to look at sub-nanometer precision inside cells and tissues. We think that this meeting will stand out because meetings in general are very specialized toward a particular technique or a particular biological problem. And here we're really trying to bridge these scales and have people come together and talk about their techniques, but really with a hindsight of how other techniques can help increase the reach of their methods. We're very excited to be joined with another meeting called Machine Learning Applied to Macromolecular Structure and Function, because AI is affecting obviously all aspects of science, but especially crazy in structural biology, as we know with the advent of AlphaFold and other technologies. And so this will really bring together through joint sessions how machine learning is informing structural biology, and the next generation of what AI will be doing is exactly working on the techniques that we have in our meeting. We brought a lot of pioneers in the field. There's going to be a lot of crazy cutting-edge science involving volume imaging, cryo-electron microscopy, live self-imaging, and all sorts of things. And also, as I said before, how they come together and get together with other techniques, and the incorporation of AlphaFold and techniques like that into all of these new technologies that are happening. I am excited about all of the program. I think that people will have a hard time even deciding on which of the two meetings there's the most interesting talk to attend. Personally, I'm very excited about, I work on cryo-electron tomography and the prospect of really bridging it to larger volume techniques. So being able to look at entire cells. And I think that there's a possibility to make all of these seamless, and we have all the right people to tell us what they're doing, and I hope there's going to be a lot of coffee breaks and drinks in discussing those aspects during the meeting. One thing that I think is super exciting is the ability for people that come from structural biology to be able to look at larger volumes and try to look at what the mechanistic insights that we get, be it purified proteins are inside the cell, but now trying to look at organelles or larger cells or really the entire volume of the cell and put in context all of the mechanism that we've learned and bring it together and apply to especially dysfunction or proteins that are related to disease. This will allow us to look at tissues and all sorts of super exciting things back and forth in resolution. I know Keystone really well. I find them incredible meetings that allow you to have time to talk to others, to make relationships, to find new fields or new ideas all the time. I've started collaborations. I've hired postdocs. It is just a fantastic meeting. That I'm really looking forward to again. Some of the best talks at a Keystone meetings are the short talks, and those are submitted by abstract. So please be one of those wonderful talks. Submit your abstract for a short talk by December 19. And if you miss that deadline or you feel that it's not ready for a talk or anything like that, you can still submit a poster on February 27th. The meetings are small, very interactive, and I'm sure you'll get a lot out of presenting your work. I think anyone who's interested in the function of proteins should attend this meeting. I think for trainees, it will be fantastic. There's a lot of pioneers in the field. For people trying to get into structural biology or imaging, if your problem requires it, or if you are an expert in one of these techniques and are really curious to jump into others. For trainees, I always think that people should try to do something different during their postdocs. It's a great opportunity for grad students. For postdocs, you get to meet a lot of people. And then for industry, of course, the value of adopting these technologies is evident. Industries are on top of it right now. And so I think that pharma should definitely come to both of the meetings, as well as other technology-oriented and other industry partners. I think that the prospect of looking at biomolecules in their natural environment, doing their thing, is coming. I think this is going to be an instrumental meeting that we're going to remember about how we talked about these things, how we connected the technologies. It's in a beautiful place. There's still going to be snow, and it's going to be a lot of fun. So please register and join us in March.